Hi everyone, and welcome to Chapter 5. So what we're going to do today is we will focus on the plasma membrane structure and function. Now, when we talk about the plasma membrane, remember that that is that phospholipid bilayer, and it has a whole bunch of proteins and lipids scattered throughout it as well. Now, again, the reason why it's phospholipid bilayer, remember the phosphates are polar, meaning they're hydrophilic, so they'll want to be on the outside where the extracellular fluid has a lot of water, and they'll want to be on the outside that's technically the inside of the cell. Okay, so if you picture this as the watery cell in the blue, the gray watery outside fluid, and it forms this double layer to make sure that the phosphates are near the water and all of the hydrophobic nonpolar tails are hidden in the membrane itself. Now, when we talk about proteins that are part of the plasma membrane as well, these proteins can either be embedded, meaning these long proteins you see here, these are called integral, meaning that they are within the entire phospholipid bilayer, so it will cross both sides, the inside and the outside layer of that membrane. Peripheral, however, if you think of peri being like perimeter, will only be on the inner surface of the membrane. Okay, so that, for instance, here where you see a surface protein, that is peripheral. This guy here, where you see it only on one side of the membrane, that is peripheral. Now, we call this membrane, the phospholipids of it, amphipathic. What that means is that's just a fancy scientific way of saying what we've been saying all along, which is that the phospholipid has a hydrophilic side and a hydrophobic side. Okay, so when you see this word amphipathic, circle it and write that it means that the molecule has a hydrophilic side and a hydrophobic side. And remind yourself, anytime you say that, what does hydrophilic mean? It tells you it's polar and likes water, dissolves in water. Hydrophobic is nonpolar and it repels water. Okay, it does not interact well. The last thing I want to mention here about the plasma membrane is the four main functions, which we've touched upon already in other chapters. The first function is that the plasma membrane separates the inside of the cell from the outside environment. Okay, so it's a barrier. It separates inside from outside. With that separation, the second function is that the plasma membrane allows transport of molecules into and out of the cell. Okay, that's where you hear it called selectively permeable, meaning it will only let certain things inside and certain things outside of the cell. It's important for transport. It's also important in communication because it will have receptors on it that allow it to communicate with other cells or molecules. Okay, so the third function is communication, and the fourth one is that the plasma membrane anchors the cell to other surfaces. So it's for attachment as well. Okay, so again, the four functions are it separates the inside of the cell from the outside environment. Second, it allows transport of molecules into and out of the cell. Third, it's involved in communication through receptors. And fourth, it's involved in anchoring or attaching the cell to surfaces. Here's a recap slide that you can pause in case you missed any of the notes I was saying in the previous slide. Now moving on, when we talk about the plasma membrane, in addition to calling it a phospholipid bilayer, we also point out that it is a fluid-mosaic model. And what you can do to help figure out what that means is break up the words. So when you see fluid-mosaic, when you think of fluids, well, think of liquids, meaning movement, right? Lots of movement, flowing. Mosaic What that means is a mixture of things. So it is a fluid mosaic because, first of all, the lipid molecules allow the membrane to be flexible, meaning it can move, it's bendy, which is why in the pictures, if you look at this picture on this slide, notice that it's wavy. That waviness that you always see in pictures of the phospholipid bilayer is to point out that it's fluid, that it's moving, swaying back and forth. And basically that's thanks to those lipids, those fatty acids that you see in the middle, which look all zigzaggy and moving. Now, the greater the concentration of those lipids, those fatty acids, the greater the flexibility, okay? More lipids means more flexible. More fatty acids, more fluid, right? The other part of the term is mosaic, which I said means mixture, and if you look at this picture of the membrane, it looks like in art when you see a mosaic or mosaic, because you have all of these proteins scattered throughout the membrane, okay? Proteins and additional lipids such as glycolipids, okay? So it's fluid because the fatty acid lipids make it flexible, and it's mosaic because it has proteins and additional lipids, even sugars, so carbs here, scattered throughout it. Okay, nice mixture. Now, when we talk about those proteins scattered throughout, there's a whole bunch of different types of proteins in the membrane, just like when we first introduced proteins and we said they're basically magical because they do everything in the cell. Well, there's 6 different types in the membranes. There are channels, carriers, cell recognition proteins, receptors, enzymatic proteins, and junction proteins. And I know it looks scary when you see them all listed out like that, but when you kind of look at the term and think about what it means in English, it'll help you think about what it means for the membrane. So first up, you have channel proteins. And when you see channels in biology, it means there's movement, flowing. Kind of think of a water channel, okay? There's flowing of molecules inside and out of the cell, okay? There's also carrier proteins, which will really bind to a molecule and help move it across the membrane. So when you think of channel proteins, a lot of times those go with gradients. Carrier proteins are more forceful, kind of like when we talk, when we talk about active movement. There are cell recognition proteins, which are like when you get sick and your cells have to fight off the pathogens. They recognize it through their membrane cell recognition proteins. There are receptor proteins, which we mentioned in the previous slides, the fact of communication between cells through the membrane, right? And they can communicate and bind to various molecules thanks to receptor proteins. Enzymatic proteins, you hear enzymes, you think metabolism, so that those proteins will help with the metabolic functions. And then lastly, you have junction proteins, and that a lot of times you see them called tight junctions. They basically help stick cells together, because remember we mentioned adherence and attachment as one of the functions of the membrane. Okay, so these are just some of the proteins you encounter. These are to help you visualize what we meant by the different proteins. So again, channel proteins letting molecules in and out of the cell. Carrier proteins being a little more forceful with the transport, physically picking up because it's moving against a gradient usually. Cell recognition proteins, so that yellow structure that you see there is called MHC, and basically that's a type of antigen, we call them, but you can think of it as a type of protein that will identify foreign invader cells, so pathogens that enter your body. There are receptor proteins, so you see that almost like a lock and key model here to help bind to specific molecules. Enzymatic proteins, so metabolism, you see it would help build things up or break things down, help things get done. And junction proteins, notice you see two plasma membranes, right? Because the junction proteins are to stick two separate cells together, okay? So when you think of plasma membranes, just please be aware of the 6 types of proteins that you can encounter, and just very briefly remind yourself what they're doing. If they're allowing for transport, if they're allowing for recognizing invaders, if they're for binding or attaching, or metabolic processes, okay? If you have any questions or concerns, you need a little review on this topic, just send me a message in Remind app. Now, when we talk about those 6 different types of proteins that you can encounter in a plasma membrane, there's one I want to spend an extra minute talking about because it's so significant in your cells, even though you may not think of it as so significant based on what we called it in the previous slide, and that's the receptor proteins. So receptors are really, really important because they're responsible for what we call cell communication and signal transduction, meaning any of the pathways in your cell that ends up for growth, for development, for making things, for breaking things down even, a lot of them depend on signal pathways, meaning they have to be told when to do certain things, told when to activate a certain gene, when to produce a certain protein. And that's done through receptor proteins. The receptor on the plasma membrane will bind a particular signaling molecule that will set off a chain event, kind of like a domino effect. And as you see in the picture, a lot of times this binding of the signal molecule and the receptor is usually in a lock and key model, which you've probably also heard before in classes talking about enzymes. And the lock and key model simply means that the signal molecule will have a particular shape and size that matches the receptor molecule. And only that signaling molecule will be able to bind to that particular receptor. Then next to it, there might be another receptor that has a slightly different shape for a different molecule. And so even with infections and stuff, when you hear that there's actually a type of receptor that's immune to HIV, there are people who cannot get HIV because their cells are not the right shape to bind that particular signaling molecule. Okay, so that's lock and key. That the signaling molecule has to be a particular shape that matches the receptor molecule. Once they bind, you then get what's called transduction. Okay, so you get the binding of a signaling molecule, which is a messenger, sets off transduction of the signal, which basically means it's a pathway of different signals passing from one protein to another to then trigger a response, which is usually in the form of activating a gene, activating an enzyme, or building a protein. And you end up getting some sort of cellular response that basically will show some form of movement or production or stopping certain things from happening. Okay, I know this is all very general, but it's just so that you can kind of get the idea of what's going on, but we're not in like biochem or A&P, so you don't have to know the specifics of the transduction. Just be aware that this does exist, that there are receptor molecules on the plasma membrane that allow for signal transduction, which is a fancy way of saying signals being passed around in the cell to get something done, to get a response. Okay? So when we talk about plasma membranes, we keep emphasizing that one of the other functions of this membrane is to control transport in and out of the cell. And remember, we said that it's selectively permeable, meaning that only certain molecules will be able to pass across this membrane. Now, there are two main categories of transport across the plasma membrane, and we're going to go into the specific examples and details one by one, but just as a general introduction, you can either have passive transport or active transport. And to help you figure out which is which, always ask yourself, what does the term mean in English? Well, if you are passive as a person, you're not, really going to let things bother you. You're not going to use much energy. Whereas if you're active, active is lots of energy, right? Bouncing off the walls, being athletic, you're active. So it's the same thing with cells. If transport is passive, it's just going to let things happen, going to let things in and out, no energy required. If it's active transport, then energy is going to be required to move things. And what really determines that, whether it's passive or active, is going to be what we call a concentration gradient. Meaning, do you have the substance that you want to move across the membrane? Is there more on the outside of that cell, or is there more on the inside of that cell? And how do you want to move them? Do you want to move them toward the higher concentration or the lower concentration? Now, in nature and biology, things naturally want to go from high to low. Okay, so high to low movement is natural, passive, doesn't take energy. If you want to go against that gradient, meaning against high to low, you want to move something from low concentrations to high, that's when energy and active transport will be required. Okay, we're going to review this with specific examples and details of each of these types of transport. So the first type of transport we have is passive transport. And a big example of that is diffusion. And when we talk about diffusion, we're talking about the movement of solutes. And what solutes are, if you remember our slides when we talked about water properties, the solute is what is dissolved or floating in the solvent, in the liquid. Okay, so in the example, when we talked about water, we said salt or sugar, that would be the solute, dissolving it then in water, or Nesquik powder in milk. The Nesquik chocolate powder is the solute. So now diffusion is moving solutes, which are those particles in the cell, and you are moving them in a way that is down a concentration gradient, meaning high to low, the natural gradient. And we call it net movement. It's net because even though you'll see molecules bouncing in both directions of the gradient, ultimately more of them are going from high concentration to low concentration. Okay? If that net ever balances out and you have an equal number going from high to low as going to low to high, then that would be reaching equilibrium, where you're not really getting any movement to change the, is there higher in one location or lower in the other. Okay? Don't worry as much about the net, the terminology of net movement or equilibrium. Focus more on the fact that when you see diffusion, it is passive, meaning it will not require energy because you are moving solutes from high to low, and that is how things like to move in biology. Now, the rate of that movement can be influenced by various different things such as temperature, pressure, electrical currents, or size of molecules. So just to help you visualize this, for instance, if you have molecules moving and the temperature is suddenly higher, well, what do you know about high temperatures in chemistry? Well, makes things bounce off the walls, right? Makes them move a lot more. So that would increase the rate of diffusion. Whereas if things are cold, kind of like if you've ever seen a fly that accidentally got into the refrigerator, when you open that fridge door again, he's barely moving. because cold stops or slows movement. So that would decrease the movement of these molecules if it's cold temperature, okay? Things like pressure can have that same effect of either speeding up, slowing down, even electrical currents. Think of if you're helping it along, helping it move, okay? And size as well. Bigger molecules, they may not move as well, okay? So again, passive transport, one big example is diffusion. You can visualize what that diffusion looks like on this slide. And so it's showing if you have the solute, which is the crystal dye, in the solvent, it's the water, okay? In this first picture, all of the crystal dye is over here. So this is high concentration here, low concentration of crystal dye or green dye everywhere else. So where is that dye going to want to move? To where the concentration is low. So you start seeing these green molecules spreading out, heading toward where there's a low concentration, fewer green molecules. And it will keep doing that until it's an equal distribution of green everywhere in that container. Okay, so there's no longer a high to go to low. It's equal everywhere. And what do we call that? That's now equilibrium. So when you have any problems focusing on diffusion, we can give you a concentration of solute on one spot and tell you the concentration of the solute in another spot. And we could ask you, well, where would the movement be? What would the final result be? And so in this example that you see here, you have 10% oxygen. So that's your solute. 10% of oxygen over here, only 2% over here. Well, in nature, things want to balance out. They want to be equal. So that 10% will keep moving to this side until it's equal. So you'll get 1% goes here, so you have 9 and 3. Well, 9 is still higher than 3. So another percent will move over. So now you're at 8 and 4. Well, this side would still have more. So then it's 7 and 5. And then 6 and 6 happens. And what do you notice about 6 and 6? They're equal. So now it's at equilibrium. Neither side is higher. So neither side will have a lower place to go to. Okay? Now there's a trick to doing these problems. So for instance, if I gave you a question on an exam and I told you that there's 10% oxygen in one half of the container and 2% oxygen in the other half, what will be the result of diffusion? Well, what you can do is you know that in the end, you want an equal amount on both sides. So what you can do, add them together. So the total in this container is 12% oxygen total. And how would it be equal? Well, cut it in half. So you take the total and divide it in half, and that gives you 12 divided in half is 6. So you would end up with 6% oxygen on the left side and 6% oxygen on the right side. And that's the same amount when we did it 1% at a time before, right? When we mapped out that the 10 would want to move 1 toward the 2%, so you had 9 and 3, okay? So if you get a diffusion problem, all you have to do is add up the percentages and divide by 2, okay? Because everything wants to reach equilibrium. If you would like more practice problems, just let me know in the Remind app, and I will give you some more problems that will match up how you'll encounter it on the first exam. Now, that kind of passive transport was focusing on the movement of solutes, meaning the particles dissolved in water. But you can also have movement of water itself. And the diffusion of water itself has a special name, which is osmosis. And so osmosis is when water moves from a high water concentration to a low water concentration. Now, if we think of that in terms of the solutes in the water, I want you to picture having a glass of water. If you have a huge mound of salt in it, right? This giant pile of salt or even sand. Picture something that's not going to dissolve. You have a giant pile of sand in that water glass. Well, you're going to not be able to fit as much water, right? When you have a whole lot of sand in there, you can only fit a small amount of water. Whereas if you have less sand in that glass, well, you can fit a whole lot more water in there, right? Same thing, even if you picture something like rocks in a bucket. Well, how much water can you throw on there? So when you think of the diffusion of water, it's going from high water to low water. If you think of that in terms of solutes, well, high water means you can fit a lot of water in there because there's very low solutes, very small amount of particles taking up space. Whereas low water is a lot of room for solutes. So that's high solutes, right? So when water is moving from a high water concentration to a low water concentration, keep in mind that the solute concentrations are the opposite, okay? So if it's high water, it's low solute. Now the reason why I emphasize that is that's important because when we talk about solutions and we ask you about the movement of the water, we actually label solutions based on their solute concentration, not their water concentration. So you have to quickly figure out whether that solution, where the water is high, where the water is low. So that's why I spent all that time talking about solutes. Because when we do osmosis problems, we ask you, well, how does the water move if a cell is in an isotonic solution, or if the cell is in hypotonic or hypertonic solutions, okay? And the way that you can visualize this, in an isotonic solution, that is the ideal. Next to isotonic solution, I want you to write happy cell. Cells are happy in isotonic solutions because there's an equal concentration of solute and water inside and outside of the cell. So there's no high or low because the solute, meaning salt and electrolytes and all of that, that's equal inside and out of the cell. So the water concentration is equal inside and out of the cell. So you don't get the cell losing or gaining any water. That's why in hospitals and medical field, a lot of times you'll hear isotonic solutions or isotonic buffers are the goal. There's also what we call hypotonic solutions. Hypo, when you hear hypo, that means low, right? So hypotonic means that there's a low salt or low solute outside of the cell, because you're talking about the solution, the environment around the cell. So hypotonic solutions means low concentration of solute, a lot of times we call it salt, low salt outside of the cell versus inside. When you hear low salt outside, what does that tell you about the water out there? Low solute means high water, right? Because if there's a small amount of salt in that glass, you have space for a whole bunch of water, right? So low or hypotonic solutions means lots of water outside of the cell versus inside. So how's that water going to want to move? If there's a lot of water outside the cell, it's going to want to come in to try and balance the water levels, right? So that cell will swell, get larger and larger until it can even burst. The trick to remembering that, when you see hypotonic, automatically tell yourself hypo-hippo. Picture a big hippo, right? Because the cell is getting bigger and bigger and bigger until it will burst. The opposite of that is a hypertonic solution. Hyper means a lot. So hypertonic means high salt solution outside the cell, okay? High solute, high salt. If there is a lot of salt around the cell, what does that tell you about the water? A lot of salt outside means there's very little water out there, right? So that means in comparison, the inside of the cell has a lot more water, okay? So the water will want to move from the high inside to the low outside, okay? So you get shrinking of the cells, okay? Think about, for hypertonic, remind yourself it's salty, and think about if you've ever eaten really salty food, like when you have really good Chinese food, very, very salty, what do you notice? Your mouth is dry because you're losing water, loses water, it dehydrates. Or really cruel kids who used to sprinkle salt on slugs to watch them lose their water and shrivel up. Okay, so this helps you visualize what we were just talking about. You have hypo, think hippo, so the water will enter the cell and the cell swells and gets bigger. Isotonic is balanced, an equal amount of solute or salt inside and out of the cell, which means equal water. You're not having any net gain. And hypertonic means more water is coming out of the cell, right? Because it's very salty outside environment, very low water. Okay, so this helps you visualize that. Okay, now I know you may think that this is getting a little repetitive, but I just want to be sure that you really get what I was saying. So just to draw out one of the examples, so hypertonic, what that would look like. So, sorry, I messed that up. Hypertonic, so that means you have a cell. Hyper means high salt, right? High salt, high solute. High salt environment. Make it all messy there. Okay, so you have the cell here. You have high salt outside of the cell. So high salt, sometimes what we'll say is, let's say 10% NaCl on the outside versus 2% NaCl on the inside. Okay, high salt tells you what? That tells you low water versus what will be inside the cell, high water. As soon as you see high water, where does the water like to move? From high to low. So it's exiting the cell. So water exits the cell. It shrinks. Okay, so again, that's how we got the idea of that movement, okay, because it's all focusing on that high to low water. We're not talking about the solute, okay? The solute movement would be diffusion. We're talking about osmosis. So even though you see high salt and low salt here, we're not talking about the salt moving. So focus on the water. The only reason we mention the salt is so that you can figure out where the high water is and where the low water is. Okay? And again, if this is still confusing at all to you, just send me a message and we will go through it in the Remind app. So this is just one more way to view how the movement of water is affecting the cells. Now, the emphasis on this slide, what we've been focusing so far is the general idea of the movement, but now I want to show you a specific difference between animal cells and plant cells. So if you remember in the previous chapters, we talked about plant cells having a very special structure that animal cells do not, and that is the central vacuole. So you'll notice that when an animal cell is in that hypotonic solution, again, hypo, think hippo, think swelling or bursting. When you see that with an animal cell, well, the more water it takes in, eventually it can lyse or burst. Plant cells don't do that. They won't burst no matter how much water they're taking in because they have the central vacuole to help store that water and help expand the cell safely. It creates what we call turgor pressure, basically meaning it allows the cell to get rigid rather than exploding. And then in the cases of hypertonic solutions where it's losing water, the animal cell will shrivel up. But here in the plant cell, the central vacuole loses water, and what happens is you get this constriction of the cytoplasm, but you may not be able to see it because of the way the outer structure of the cell is. Okay, but this is just to show you what we meant in the previous lectures when we talked about the central vacuole. The next type of transport that I want to talk to you about is facilitated transport. This is another example of passive transport, and when you see passive, that tells you two things about this type of transport. It tells you that facilitated transport does not require energy, so there's no energy here. The second thing it tells you is that the solutes, or the molecules that are moving, are going with the concentration gradient, meaning they will be moving from high concentration to low. So wherever there's a higher concentration of the solute, they will cross the carrier protein to get to the lower concentration. Now, the only way that this differs from the previous diffusion that we talked about is that there is a carrier protein involved. And that carrier protein will be specific to the type of solute that you want to move. So in this case, it might be glucose. So only the glucose molecules will get into that carrier protein and move into the cell. Okay, so it's a way of regulating the movement into and out of the cell, but it's still going from high concentration to low concentration, so no energy is required. Okay, now keep in mind that once again, we are talking about a form of diffusion again, so we are focusing on the solutes moving. Only in osmosis were we talking about the water moving. Now, in addition to passive transport, there are also a lot of solutes that have to move based on active transport. And as soon as you see active transport, I want you to write down requires energy. And it specifically requires energy in the form of ATP, that nucleic acid molecule that we mentioned in the previous chapters. Now, why does it require energy? Because you're moving things against the gradient. Okay, so as soon as you see active transport, think energy, because we're moving against the gradient. And if that natural gradient is high to low, that means that active transport is moving solutes from low concentration of solute to a high concentration of solute. Okay, so for instance, if the solute is low outside of the cell, high inside of the cell, well, you would be moving more of them to the outside, even though there's already a lot there. Things don't naturally want to do that, so that's why it requires energy. And it will usually use carrier proteins, such as you see in this picture here. These carrier proteins help it move across the membrane, again, because it's not natural movement. So you gotta help it along with energy and a carrier protein. You also, across the membrane, have what's called bulk transport, and this is very, very important type of transport, and you actually saw it in a previous lecture, but we didn't refer to it as bulk transport. You saw it when we went over the Golgi apparatus. So remember that the Golgi was the packaging plant of the cell, and it put those modified lipids and proteins into nice little vesicles, and then pushed them out of the cell, secreted them out. Well, that's an example of bulk transport. That is exocytosis. And when you hear ex or exo, it's on the outside, okay? Exo, like exoskeleton of insects, it's on their outside. So exocytosis is when a cell is releasing contents. So it's releasing lipids or proteins or digested materials. It's excreting them. So if you see the word exocytosis, you can think of cell excretion, getting things out. Whereas endocytosis, endo means inside. So endocytosis means that the cell is taking something in. And it can either be in the form of phagocytosis or pinocytosis. And phagocytosis is cell eating. Okay, in phagocytosis, the cell will engulf large amounts of solid material. So for instance, when you have bacteria invading your body, it's a pathogen, some of your immune cells will be phagocytes, meaning they will engulf that bad bacteria and break it down and digest it for you. Okay? And then after endocytosis, what will it then do with that broken down dead bacteria? It will perform exocytosis to excrete it out of the cell. Okay, so they're tied together. Think of phagocytosis as cell eating, and then exocytosis is cell pooping. You also have pinocytosis. Okay, think like pina colada or Pinot Grigio. Okay, pinocytosis is cell drinking. So in this case, the cell will engulf liquid or really, really small solid material that's practically liquid. Okay, so that would be kind of like a smoothie. So pinocytosis is cell drinking, and there is one specific example or specific form that some cells use, which is called receptor-mediated endocytosis, which is a form of pinocytosis, so it's taking in liquid. And in this case, it's using a specific protein that will bind to small particles in the liquid. So it's a more specific form of receptor-mediated, a more specific form of pinocytosis. Think about it as someone who's lactose intolerant, so they will only want to drink soy milk or almond milk. So they need receptors specific to that because they don't want to take in regular milk. Okay, so make sure you're comfortable with what the terms exo and endocytosis mean, the difference between phagocytosis and pinocytosis. And keep in mind, receptor-mediated endocytosis is just a specific type of cell drinking. This is just a visual, and again, you've seen this in a previous chapter when we talked about the Golgi apparatus. You saw the Golgi having a vesicle formed and then merging it with the cell membrane to spit out the inner contents. And again, if you want to think of this as cell pooping, you can. Then the opposite of exocytosis, these are the visuals of endocytosis. So now instead of pooping out the material, it's taking it in. And again, phagocytosis, you're taking in large solid molecules such as a whole bacteria as the cell engulfs it and then will break it down. Or pinocytosis, little tiny particles basically taking in liquid or a smoothie, as you can think of it, into the cell. Receptor-mediated endocytosis is taking in very specific tiny solutes or liquid. Now, one of the last things I want to mention is with the membranes, there are sometimes important extracellular structures to be aware of. In animal cells, whenever we talk about the extracellular matrix, we're basically talking about this mesh of proteins and sugars, carbohydrates on the outside of the cell, so directly outside of the cell. And a lot of times it's made up of things such as collagen. So if you've heard of collagen in your skin, for instance, That's something that resists stretching. Elastin is a very resilient support structure. And fibronectins, you also see those scattered throughout. Sorry, there's the highlighter. So it's scattered throughout here, that green particle there. That's basically binding to the integral proteins. So remember we mentioned integral proteins are the ones that go completely across the membrane, both sides of the membrane, embedded within. The fibronectin will bind and interact to those integral proteins to basically assist in communication between the outside extracellular matrix and the inside cytoskeleton of the cell. And the reason why this communication is important is it helps with movement of those cells with that outside world and being able to attach and move. Now, when we talk about the attachment and movement with the outside world, there are three structures I want you to be aware of. We call them junctions between cells. You have adhesion junctions, tight junctions, which I mentioned earlier, and you have gap junctions. Each one has its own kind of function, and you can visualize the differences between them in these figures below. So adhesion junctions, as they state, are for adhesion, meaning it's for attachment. So these will physically attach two cells together. And they do that with these intercellular protein filaments that you see stretching between them. So adhesion junctions are for the physical attachment of two cells. Tight junctions are also for the physical attachment to two cells, but they're very tight, and we call them impermeably connected, meaning you're not going to have movement between those two cells. They're tightly connected. You notice you don't have a gap between them for transport of molecules to get through. You then lastly have what are called gap junctions, and they look almost like the carrier proteins and the channels we saw in earlier slides. These are basically meant for communication between cells. So gap junctions are for joining cells together, but allowing for the molecules and ions to be exchanged between those two cells. Okay, so you have loose physical connection, you have tight physical connection for attachment. Both of these first two are for attachment, whereas the third one is for communication between cells. Then the last structure I want to mention is specific to plant cells. So with plant cells on their outside of their membrane, they have the cell wall, which we mentioned. Animal cells, such as our cells, do not have a cell wall. Plant cell walls are important for them because they are extra protection and they allow for nice, free kind of specific movement of molecules and structures into and out of the cell, just like the plasma membrane would. Plant cell walls are mainly made up of cellulose, and I want you to circle star, highlight the term cellulose, and remind yourself that in a previous lesson, we focus on cellulose as being an example of a carbohydrate. Okay, so remind yourself that cellulose is a carbohydrate, and its function in plant cells is for structure. It gives rigid structure. And remember, we cannot digest that. So when you eat plant products, you're only able to digest the cellulose because you have bacteria in your gut helping digest it for you with their own enzymes. Then the last structure I want to mention are the plasmodesmata. And plasmodesmata in the figure here, that's these structures here where you see the arrows going in and out. These are plasmodesmata. They are channels that pass between the cells, and they allow for the exchange of some materials from adjacent cells. And that's very, very important in plants because what do plants have to move amongst their cells? Water. So plasmodesmata are how cells, plant cells specifically, get water carried through the entire plant. So when I say what do plasmodesmata do? They're channels in plant cells that allow for water to get carried through the entire plant. And again, remember the way that that water gets carried through is through cohesion with other water molecules and adhesion with the surfaces of the plant cells and tissues. Okay, so as you go through more and more chapters, keep reminding yourselves of what you've learned about this stuff in previous chapters because it all comes back. Okay, now that we've finished yet another chapter, at the end of every chapter, always remind yourself that you have to add this chapter's material into your weekly study guide, and you may want to use some of the information in this chapter to come up with questions for your weekly discussion board. Okay, that's it for Chapter 5. Any questions or if you want any practice problems, just contact me in the Remind app, and have a wonderful day.